All right, we have one last talk that's going to be from Anuj Karpatne of Virginia Polytechnic Institute and State University. He'll be talking about biology-guided neural networks, integrating biological knowledge with neural networks for discovering phenotypic traits from fish images.
Hello, everyone. Can you hear me?
Yep, your slides look good too.
All right, perfect. All right, thank you so much for inviting me to this session. This is a very exciting workshop. My name is Anuj Karpatne. I'm an assistant professor in computer science at Virginia Tech. And today I'll be talking about a very recent line of work that we're exploring in this rich world of kgml research, which is building a new class of biology-guided neural networks. So in this workshop, we have had so many great talks discussing several types of scientific knowledge that is being ingested in machine learning frameworks. We have process-based simulation models. We have partial differential equations that have been discussed. And a lot of talks have focused on time series data. This talk would take a complementary perspective, adding to the diversity of kgml research by talking about image data and how we can leverage scientific knowledge available in a very different form, in the form of knowledge graphs, ontologies, and phylogenies that we have available in biological sciences to improve the generalizability, robustness to adversarial attacks, even in the paucity of labeled data. So this is connected to aquatic sciences because the focus of this work on building biology-guided neural networks, or BGNNs, is to understand and study fish images better. So we are interested in studying teleost fishes. And in my talk, I'll talk about the high-level objectives that we are exploring here. My objective would be to see similar ideas being explored in other problems of aquatic sciences. Yeah, okay, I got my mouse pointer back. Okay, so let me first introduce you to the team behind the BGNN work. This is a collaborative project with 5 PIs and more than 5 institutions that are coming together. This work is funded as part of the HDR Ideas Lab activity. So all of this work started in 2019. Thanks to NSF, we all came together as part of a week-long activity from very different disciplines, where we thought about a really interesting scientific problem that we could solve by harnessing the data. In this case, we looked at harnessing image data and how we can leverage existing scientific knowledge for this problem in the spirit of kgml research. So this has led to a very interesting project. We are learning a lot from collaborating with biologists and computer scientists in our team. And I'll talk about some of the research highlights that we have experienced in this project. So let me start with the high-level vision of our BGNN work. So in many biological problems, we are working with 2 separate compartments of resources or knowledge, right? On one side, we have resources available in the form of image data, which is growing by the order of millions in many biological applications. For example, in biodiversity research, we have so much of images that are being collected from different sources. We have data that is being collected from museum and university library collections. For example, IDIC-Bio, GLIN. These are established networks of recording data in the form of images. And we also have a lot of excitement in the community on generating citizen science datasets. So these are crowdsourced datasets, for example, collected by tourists posting on social media through camera traps, through drones. So there is a vast richness of images, and it rightfully so. Images are now being considered as the new currency for biodiversity research. Right? So this is one source of knowledge that we have in some sense in biological problems. Our focus in the BGNN project is on understanding teleost fishes. The other compartment is the availability of existing scientific knowledge about teleost fishes, right? So that this knowledge is available in the form of different types of relationships between biological entities. For example, for teleost fishes, we have understanding of the phenotypic traits, for example, the different types of fins, and their properties that help distinguish between the different species. Right? We have knowledge about how the species are related to each other from the tree of life, from phylogenies. We have relationships between traits and species, and we have relationships between traits. Right? And many a times, scientific knowledge in biology is expressed in the form of ontology. So here is just one example of an anatomy ontology. Right? So a lot of research is happening in collecting more image data and in discovering new scientific knowledge, right? But these are the two compartments that have siloed in some sense. And there is an opportunity to bridge the gap between these two silos. So for example, given all of these images, would it not be interesting if we could automatically extract new scientific knowledge from these images, right? So we have a lot of trade data that is trapped or captured, locked in these images. What if we could unlock them and discover new relationships between the species, between traits and species, or between the traits? This is the high-level goal of our project on BGNN. We want to build machine learning models that can automatically extract new knowledge, harping on advances in deep learning that have revolutionized the field of computer vision, working with benchmark data sets, having millions or even more labeled images. Can we harness those recent advances in deep learning for a number of machine learning tasks, for example, for classifying species, for detecting traits, and even to segment the traits given these images, right? Once we are able to do that, it would lead to a lot of new knowledge, new insights that we would discover that is currently locked in these unlabeled large collections of images, right? So to solve this grand challenge, the black box machine learning methods are unsuitable for several reasons. And these reasons are very similar to other research directions that are being explored in kgml2. So the first challenge that we experience is the extreme paucity of labels that we have for many of these machine learning tasks. For example, if we talk about the problem of trait segmentation, we have more than 100,000 images that we are currently working from a museum collection data set. And through our efforts, we have been able to annotate about 500 images out of these about 100,000 images, because labeling the traits in these images requires significant domain expertise, right? And it's time and labor intensive. Now, this is something that is in stark contrast of the scale of labels that we have in many benchmark computer vision problems that have been one of the key drivers for advances in deep learning. So in the paucity of labels, it's easy for a machine learning method to show poor generalization performance. Another major challenge that compounds this problem is the poor quality of data of images that we have in many real world applications. For example, if we talk about museum collection images, which are still relatively more standardized than crowdsourced images, images collected in the wild, we still have a lot of challenges in dealing with several types of artifacts. For example, occlusions of traits. For example, a fin of a particular fish may be folded, or maybe the entire fish is folded, or maybe the image was taken vertically, not horizontally. Right? Now, all of these data quality issues further exacerbate the problem of poor generalization performance of black box machine learning methods, that in the presence of these quality issues are susceptible to making spurious decisions. So this is the basic context driving our research in the spirit of KGML research, which is how can we augment black box machine learning methods using scientific knowledge available in this particular domain in the form of phylogenies and ontologies. And this is what we are referring to as the family of biology guided neural networks, or BGNNs. So in this talk, I'll talk about three very simple problems and how we are leveraging scientific knowledge for these problems. So the first problem that we have explored is of species classification. So we are given an image of a fish, and we can run it through a convolutional neural network that can express a hierarchy of hidden features at the layers to finally predict the posterior probabilities of this image belonging to one of the species class. And again, for this problem, the major challenge is the paucity of labels. Right? So, for example, from the university collection databases of images, more than about 450 out of 575 species have fewer than 30 images per species. Right? So we have very small amounts of data for most of the species. So to solve this problem, the type of biological knowledge that we are bringing to the table is relationships between the species expressed in the form of taxonomic or phylogenetic groupings. Right? So the key idea here is that if we want to predict the species class, we know that the species classes are related to each other. Right? And this relationship is expressed in the form of phylogenetic trees or taxonomic groupings. For example, two species that share the same genus can be expected to share similar neural network features at its hidden layers. Right? So if I look at two different species belonging to the Neutropis genus, and if I take these two images and run it through a neural network, we would expect that these two species should express similar features at the hidden layers as opposed to images of species that belong to two different genus. Right? So this is the basic intuition that we want to bring in, which is the species-species relationship. Okay? And the ultimate goal here is to bridge the gap between the hierarchy of features that we are learning in deep learning models with known hierarchies in biology, for example, taxonomic groupings and phylogenies. Right? So this is the basis of a line of work that we are referring to as hierarchy guided neural networks, where the hierarchy is coming from biological knowledge. And there are several ways to express this hierarchy. We present a very simple approach that has recently been accepted at Methods in Ecology and Evolution. So given any image of a fish, we can run it through two blocks of neural network layers. We can have some species features that help in predicting the species probabilities, and we can have another block that predicts the genus probabilities, having its own set of genus features. Right? And for example, we can build two different convolutional neural network blocks for these two problems. Right? But we know that these two problems are related. Essentially, if we want to predict the species probabilities, we know that the features that we have already learned for predicting the right genus are useful to predict the right species, right? So this is informing the architecture of the top convolutional neural network that is trying to predict the species probabilities using features expressed for a coarser scale problem of predicting the right genus. Right? So that this is a very simple idea of using genus level features for the problem of species classification. There are many more involved approaches that we are currently exploring using phylogenetic information rather than taxonomic groupings. But even this very simple idea is able to demonstrate that by using biological knowledge, we can obtain better accuracy, especially when the amount of labels is small, used for training, and especially when we know that the problem is hard. So here is a graph where we have data coming from two different data sets that we call as easy and hard. The easy one is obtained from a single museum and has a relatively simple collection of species and genre, whereas the hard has images from 5 different museums, and it has much more diversity in this set of species. What we can see is that for the hard data set, HGNN is able to show better accuracy. And for the easy data set, when we starve the data set with smaller number of images per species, that is when the power of bringing scientific knowledge in the form of HGNN really outperforms black box models. So this is just one line of evidence. There are many other ways to look at the performance of machine learning models. For example, by looking at saliency maps, which are essentially regions of the image that a neural network finds to be very interesting in solving the downstream machine learning task. So we'll be looking at these saliency maps and the species probabilities that we are predicting to look at a very interesting insight that we obtained, that by bringing in scientific knowledge, we can even achieve better robustness to adversarial attacks. Right? So here is a very simple adversarial occlusion, adversarial attack procedure that we explored, which is by occluding regions of the image that have the highest contribution to the saliency maps. Right? So for example, in this particular image, we are given the saliency map, and we are given the probabilities predicted by a black box model in HGNN. We know that the first class here is the correct class, right? So we can see that both these models are doing fairly well. But what if I put a patch on the nose of this particular fish, which had the highest contribution to the saliency map, right? So we are essentially starving information, right, by occluding certain regions that the neural network finds most informative. Right? We can see that both these models take a hit, but the hit by the HGNN model is much smaller in predicting the right class. And we can repeat this procedure several times, and we can see that in just 2 steps, the black box model is easily thrown off. The highest probability that it is predicting is for the 3rd class, which is incorrect. So this shows that by anchoring machine learning models with scientific knowledge, we can even be more robust to these occlusions that right now we are simulating adversarially, but can happen naturally in real world images because of the poor quality artifacts that we discussed in the beginning. Let me talk about another problem where we are looking at the problem of detecting traits. So given an image, we want to perform a multi-label classification of all of the different traits that are present or absent in this particular image. The challenge, of course, here is that we do not have trait labels at the level of individual specimens. Right? And this is where we can use another form of scientific knowledge, which is in the form of species trait occurrence matrices that are already provided to us by a domain expert. So for every species, we know which of the traits are present or absent in general for that particular species. This, of course, is known to be highly incomplete, but that's the exercise that we are currently working on to perform trait detection and even segmentation using species trait matrix and a small amount of high resolution trait segment labels. So in the interest of time, let me talk about a third application that we are exploring, which is to perform semantic segmentation of these images. So given an image of a fish, we want to identify all the interesting traits in the image. We would again face the challenge of paucity of training labels as we discussed in the beginning. So here is where we are exploring scientific knowledge available in the form of trait relationships. Right? So let me, in the interest of time, skip over this cartoon example and talk directly about the type of relationships that we are exploring here, which is for all of the traits that a neural network model is predicting on an unlabeled image, we can enforce whether those traits satisfy known spatial relationships between the traits. For example, we know that the pelvic fin is anterior to the caudal fin. Right? For any unlabeled image, if a machine learning model violates any of these relationships, then I know that I need to go back and retrain my model. So this is an ongoing work where we are making use of large volumes of unlabeled images and checking for consistency by querying anatomy ontologies and backpropagating that in our segmentation model. So just to summarize, this project explores a very interesting line of research in using a very diverse form of scientific knowledge available as ontologies. This adds to the richness of the ecosystem in kgml research, and there are several research needs that we need to explore going forward for kgml in aquatic sciences. So I would like to thank the entire BGNN team and the funding agencies and my wonderful group of students. Thank you.